Hello everyone, this is Group 2. I'm Minji Yu and here's Chaeyoung, Yewon, and Seungchan. This is the presentation of our project manufacturing the biosimilar of Dupixent. These are the contents of our presentation. At first, I will introduce the product that we will manufacture. Our original product for manufacturing biosimilars is Dupixent. It is the #1 treatment for atopic dermatitis, aka PPD in Korean. It has been developed by Sanofi Regeneration, a pharmaceutical company in France. The patent's expiration date is 2028 in US, 2035 in South Korea. So the original cost of the product is $1,500 US. Considering the general guideline of Dupixent, the year cost is $2,600 US per person, which is quite a huge cost for individuals. Therefore, we decided to develop biosimilar for Dupixent to reduce the market price and earn sales. Next is about the target market. The figure on the left is the trend line of sales of Dupixent from 2017 to 2022. As you can see, it is currently increasing with average growth rate of 105.2%. And also looking at the sales distribution of Dupixent worldwide on the right figure, US took the largest proportion, about 77%. Therefore, we decided that developing Dupixent biosimilar can make money, and then targeting US market is necessary. With the production forecasts, the left one is our prediction of US sales of Dupixent in 2028. This was calculated by multiplying sales and growth rate and the proportion of US sales market. Then we found the information provided by Evaluate Pharma that predicts $18 billion sales of Sanofi due to Dupixent in 2028. Since this is more credible than our own prediction, we used this number to get our target file. With the market share as 20% and sales price of $525 for the competitive price, the target vial became 5,259,428 vials until 2028. And here's the concept. Since the product is too expensive for individuals, we are trying to make a biosimilar of the Dupixent. Considering the manufacturing process, we tried to use CMO, but the problem was that we should operate the CMO factory for a whole year 24/7 to make our targeted vials. Therefore, considering the mass production in short periods, we selected to remodel the existing factory. Finally, comparing single-use and multi-use, with low initial cost and high time efficiency for mass production and avoiding cross-contamination, we decided to work on single-use equipment.
So let's talk about process now. This is our BFD, a typical process to manufacture monoclonal antibody. On upstream process, we designed the process to minimize steps. It is because less steps lead to the cost reduction when it comes to introduce new equipment. So we only operate one locking incubator and one bioreactor with the intensified cell concentration. As a result, the total upstream process takes 28 days, and the final concentration of antibody is 5.5 gram per liter. At the downstream, antibody is clarified by centrifugation and depth filtering. Then the clarified media is mixed with 20 liter of 50% of polysorbate 80 to match the final concentration to 0.4% in the solution. After that, protein A chromatography is executed. The media first flows through and 5 column volume of elute buffer is put into the column for 5 hours. Through three steps, 8,616 gram of antibodies are remain for next process. Next, IEX and virus filtration is executed. The eluted antibody solution flows through the column of anion exchange chromatography column. 10 column volume of start buffer and 10 column volume of elution buffer are spent. It takes 7 hours. After that, the elute passes through the virus filter to remove virus. 2 hours is spent in this step, and in the end, 8,018 gram of antibody is remained. After polishing steps, the solvent of antibody solution changed to the 25 liter of buffer for 7 hours. Again, after it finishes, the solution passed through the filter again for 2 hours. The antibody is packed into single bag and then stored. Later, it is stored and packaged into with 0.6 additives. The final antibody is earned with 7,540 grams.
Let's move on to the process mass balance part. 1820 liters of culture medium and 900 liters and 19 liters of each supplementary medium are spent in upstream. The cell grows up to 5 times 10 power 6 cells per 1 milliliter, ending up with 11,000 of antibodies per 22,000 liters. Among them, 85% of antibodies is only viable. In downstream, numerous solutions are spent for polishing. In some, 1070 liters of ultra-pure water is spent. 69% of antibody from upstream remains after this downstream process. The table in this slide indicates the yield and volume recovery for each step. This slide is about scheduling. We first calculated the target amount of antibodies. From the introduction part, our target vials per year were 5,259,428 vials. As a vial contains 0.03 grams of antibodies, we got our target amount of antibodies 157,783 grams. And from the process mass balance table, total antibody from a batch, which is 7540 grams per batch. We should produce 21 batches for a year. Using the result we got from the previous slide, we made a schedule by using a 1 rocking incubator system, 2 bioreactor system, and one downstream system to save our initial investment cost. We decided to make 2 batches a month. To meet our needs for a year, about 11 months are taken. Our factory should be on operation for a whole year except for 1 month.
So this part is about marketability. This is our equipment and cost estimation. Our project consists of 2 upstream lines and 1 downstream line, and we utilize main products from the company Cytiva in the upstream process. Cytiva was very helpful in providing machine prices when we reached out to them, which made price estimation easier for us. The single-use products are marked in yellow. And for the downstream process, we mostly used equipment from the company Sartorius. And the capex was estimated by adding up the cost of all the equipment, excluding the single-use products, the factory side, the construction cost, and the value-added tax. The total amount came out to be approximately 50,430,000. And the OPEX was calculated to be approximately 58,370,000. As a result, our ROI value is 258.9%, and the payback period is 0.027. We think this is too unrealistic, and we think there is a problem with our pricing. So if we are working on this project, of course, let's go, but we need to measure the price realistically once again. And this is our Gantt chart. We proceeded with this project after a total of eight weeks, excluding the 2 weeks of the exam period. After the project, our members talked about what was difficult and what was disappointing. The most talked about was that it was difficult to measure the price of the equipment because it was confidential information and we were not sure if we were doing well. Therefore, it took a lot of time. And we talked a lot about the need for adjustment in pricing. We also regretted that we didn't consider equipment compatibility for each company. And this is individual feedback of our group members. And these are our emails and our references. And thank you for listening. And this is the end of our presentation.